normal patients. So that's where I came up with some kind of this thing. Why don't we apply look for the efficacy of the bacterphos therapy in the diabetic condition? So that's where it started. In the beginning, Chandrakant Kevani sir said, I have a lab, you do whatever you want. So I started a glutamine synthesis project. I went to Indian Institute of Science. I brought so many literature publications. But there was an interest towards this bacterphos therapy. After a period of months, I stopped that. I moved into the bacterphos therapy work. There was a lot of struggle. So this is how it started. When I decided, I said, sir, I will continue bacterphos therapy. Then I started isolating the bacterial Staphylococcus. For that, I took madam's colleagues, friends' help. Dr. Rajeshwari in MRMC college. So I approached her, we got some isolates. In a period of some 3 months, I got some isolates. That's where I started gathering the Staphylococcus isolates for continuing the bacteriophores. Then, in the meantime, Dr. Sripad A. who came for my MSc dissertation, he was interested in the bacteriophores therapy project. So I continued the discussion. That's when we started collaboration or program. There I got a help for electron microscopy and ELISA work in advance. Then MCCS, most of our colleagues, we used to do 16S RNA sequencing for confirmation of the isolates which we isolated from the medical. That's from Kurdish Chochi in MCCS. Then when I was reading about MRSA, I used to always see one center in Bangalore, Tata Saurabh Center next to IAC. Gayatri is an eminent professor. She worked on MRSA. So I wrote an email to her. I have some Staphylococcus isolates. I want to do a PhD. And I explained about bacterial therapy and all. She was interested. She invited, she allowed me to do a PhD in her lab. That's where I went to her lab for a week. I performed PhD for these isolates which we gathered from MRMC college. Then comes conversion BIOTECHNOLOgy. Which is the only company working on bacteriophores in India. When I wrote to Professor Bharti Sriram, she is the president of the company, expressing my interest in developing bacteriophores therapy for diabetic condition. She was so much interested and she helped in characterizing and enriching the bacteriophores which we isolated here because we were having a problem enriching that thing. That's where I got conversion help for enriching my bacteriophores, characterizing that thing. Then comes Navadaya Medical College, that's Dr. Chandrakant Kevani's friend, Dr. Srinivas. With his help, I did bacteriophore therapy testing in the mice because Department did not have an animal facility. In Raichur, for a period of 2 months or 3 months, I stayed there and we performed this animal studies. Then I took a help from Dr. Nimbati sir and Dr. Sivana sir from microbiology department to review my manuscripts to publish those articles. So with all this collaboration, I was able to publish at least half a dozen publications from my thesis with a more than currently cumulative with a more than 500 citations. All these publications are in most of international journals. And there was a overseas fellowship for me from Dr. Nelson, Daniel Nelson Labs. Because of some family reasons, I could not take it up. And later, our sir got the opportunity to have that overseas fellowship and he visited his lab. Then what happened, the part which we isolated in our lab, that was deposited in the VA resources in US. So the first GRCS, which is currently available in BI resource, which is maintained by ADCC. G stands for Gulbarga conversion, the company which helps for isolation. R stands for Raju, C stands for Chandrakant, S stands for Staphylococcus. So this is the pause that nomenclature that we did, and currently this pause has been characterized, and it has 2 to 3 publications on the daily medicine. So the reason, the combination of this one slide shows that if you have some ideas in your mind and if you put them together with the thought process and collaborative modes and with the advantages of this existing other things, we can literally make it possible. So with this work, we moved from that work to, that thesis which allowed me to travel all in most of the international reputed institutes, including Indian Institute of Science, QMR, Brisbane, Australia, then Albany Medical Center, USA, and ICG, Italy. Of course, now I'm working in industry setup. So earlier my organization BIOTECH and Indian University. So this I thought kind of motivational slides for the youngsters who can think that there is no much facility how to do PhD and all. This collaborative things works whether it is in a state university or central university or even in institutes. So with this, I would like to move on to my experience, which I worked in earlier organization, where I was part of a program called intranasal vaccine development. That was the world's first intranasal vaccine for the COVID-19 vaccine. It was developed by Bharat. I think all of you know about Covaxin, which was a successful story of Bharat. And along with Covaxin, Bharat also has a second program called IncoVac. Because of my earlier experience in USA where I worked with the BSF Capital, I got an opportunity to lead this project. And we have done all characterization, preclinical immunogenicity studies of this vaccine. And the biggest challenge for the doctors and medical healthcare workers is how to administer this vaccine into people, because everybody is familiar with the injections or oral administrations. Nobody is, having experience of nasal administration. So I was training lot of healthcare workers how to administer this vaccine. If at all this, of course, this launched and there were several people took this vaccine, but by the time COVID subsided. Next slide. So my current organization, which is Indian Immunological Limited, which is a subsidiary company of National Dairy Development Board, NDDB. It is established in 1982. It's almost a 4.5-decade-old company, and it produces vaccines for both humans and animals. Actually, it started in animal vaccines. Then in 2000, we started the human vaccines. We are the largest manufacturer of FMDD. We have more than 2,000 employees with the BSF IVL as a One Health approach. That means we are concentrating on vaccine development for humans and animals as well as the environment. That's where you can see our portfolio. We have vaccines for humans, cattle, goat, sheep, pigs, cats, and now we are the first manufacturers for getting into the vaccine development for fish animal. So that way, IL is developing vaccines for human health approach. And we do have vaccines which falls into all categories, namely inactivated, live activated, subunit vaccines, recombinant vaccines, adenovirus-based vaccines. I think you all are familiar with these vaccines. And currently, we are also taking the pre-concept mRNA vaccines. Coming back to the vaccine development, this image shows how much importance of vaccination in the current scenario. So in 1986, in a span of 1 year, infants used to get maximum 4 to 5 shots of vaccines. But if you look at in 2026, the number of vaccines includes 4-fold. That means there is a huge demand for vaccine development, and there is another side, there is an increase in the population. That means there is a heavy challenge for manufacturer how to bring the vaccines into the market, how to scale up the existing vaccines to fulfill the existing demand. So with this, I would like to bring into the basics of vaccine development, which the youngsters can understand. See, vaccine development has basically 4 to 5 stages. First one is vaccine development, second one is preclinical trials, then comes clinical trials, government renewal, approval, then comes the quality. So these are the stages vaccines go through whenever there is a demand for a new vaccine development. So if you look at the malaria vaccine, the question comes, why does it take so long to develop a vaccine? The vaccine program started in 1960, they got approval in 2020. That means 62 years. Of course, in the first 15 years, they had a different approach of X-ray activated vaccine. But actual antigen they discovered in 1983. 1984, the research started. From 1984 to 22 is also a decade of research, which went into the development of malaria vaccine to bring that vaccine into the market. So you can understand that vaccine development is not an easy task. Okay, this is the scenario with malaria vaccine development. When it comes to COVID-19 vaccine development, we developed, or globally the vaccines were developed next slide. In a less than 1 year, BIOTECH developed an inactivated COVID-19 vaccine. How that is possible? So if you look at the scenario, for malaria vaccine it took 4 decades, when it comes to COVID-19 it took less than a year. So it clearly says that when there is a pandemic, when there is a global issue, things are different. Malaria is confined to developing countries like African countries and South Asian countries, things are different. Okay, so if you literally look at the timeline, the vaccine was isolated and characterized in May 2020 by NIV, but Buras is here. He was a part of that program. Okay, it was characterized or isolated in Jubilee. May BIOTECH received and they got approval in 2020. Less than 1 year. So if you look at the reasons why COVID-19 vaccines were developed so fast, which one is real? That the malaria vaccine development pathway is real or COVID-19 pathway is real? Up to some extent, I would say malaria vaccine development vaccine is actually, most of the vaccines goes through that pathway. Because of this pandemic, because of urgency, because of the emergency throughout the globe, sometimes there will be a different channel without compromising with the quality or safety aspects. Here what happens, there was a pre-existing data platform for COVID-19 vaccine, because SARS-CoV-19 is also a family of SARS, which has already, there was outbreaks. So there was some work was established on that. So that pre-existing information was utilized to come up with, they did not spend much time on antigen identification and upscale manufacturing. Industries took the risk of going ahead and making the batches, even if the vaccine fails, they were ready to face the loss. Then comes adapted regulatory authority. Apart from the manufacturer, regulators also work tirelessly around the clock. They enhanced or they did a overlapping studies, approvals. That way what happened, there was a close collaboration, manufacturers and the regulators as well as the research institutes. With all this, of course there was a funding from funding agencies. So with all this, the COVID-19 vaccine was able to develop in a span of 1 year. So, as Nata was mentioning that any vaccines or any biological material, if you want to use it from human-derived biological material, if you want to use it for research purpose, you need to have a regulatory approval. So when it comes to vaccine development, the question is, do we need a regulatory approach? Yes, you need to take approval from IAC, RCGM, GAC, SEC, BCGC, so many regulatory approvals you have to take at every stage when there is a progress in the vaccine. Next one. So Institute Biosafety Committee, I think in university most of us are familiar with this. Whenever I want to start a new research with any reagents or any bacteria, virus, strains, or anything, I need to take IBC approval. Basically, that IBC committee will review the protocol, risk assessment, and safety of that reagent, and there will be a composition of that. So this IBC is must for starting the project. So this is where a key project vaccine development will start. Same thing will apply to industry as well. In industry, where we work in research and development, we do need to have a IBC committee. We need to get our IBC approval for every project, every new reagent which we are going to use for our project. So if the project involves recombinant antigen, genetically modified pathogens, then apart from IVAC, we also need to take the RCGM approach, that is must for especially the BSL3 and recombinant pathogens. So antigen selection and vaccine development. This is the preliminary or basic approach. This is where it starts. For example, COVID-19, everybody started looking for the antigen, which is the protective antigen of the pathogen, so that I can use that antigen as a protective approach to develop the vaccine. So this takes some time where you basically need to find out antigen development that should be immunogenic and that should be common across the serotypes. It's not like if you develop a COVID vaccine for one state, if it is not going to work for other states, then it will be research. And it should elicit both B-cell and T-cell response. That means whenever any research involves for vaccine development, they need to take care of these things. It should be immunogenic, it should be common antigen for all the serotypes, it should elicit both T-cell and B-cell response approach. It should not cause autoimmune responses. Okay. Then comes most of the vaccine development happens with the H2s. Industry less likely to get into the area. What we do is we get a technology transfer from the H2, like universities or national institutes or international institutes. They will use that particular code which is expressing immunogenic antigen. Then comes a vaccine development, key elements which happens in the industry. We need to first develop a process. That process should be, very optimal, and it should work from flask to 1,000 liters of reactors, and then comes upstream. Upstream means expansion of the initial activities. Then comes in-process testing. At every stage, we need to test antigens, antigen determinants, quantification of the antigen. All this is to develop across the space. Then comes the formulation, downstream. Means once you have, for example, if you are expressing an antigen in the cells, viral cells, except the protein of interest, there should not be any impurities which comes from the viral cells. That has to be taken care by process development. So you need to show that at the end you have protein of interest, not any other impurities. That impurities within the specific limitations. Then comes the formulations. The formulation is very important. Make sure that that antigen is immunogenic and it is stimulating both TH1 and TH2 response which is required, and also duration of immunity is also important. That duration of immunity you can get only when you have a potential achievement. So after doing this, now we want to test this vaccine first with the animals. To perform animal studies, you need IAEC approval, that is Institute Animal Ethics Committee, which is very important before you start the studies. So this approval is must to test your vaccine in the laboratory animal models. Here it comes for preclinical testing. So preclinical testing, what we do is we will do the different concentration of antigens, dose escalations, and we test which dose works, which formulation works and all. So finally when I freeze my antigen concentration and adjuvant, then I will say this is the formulation which I am going to use it for my clinical trial. So clinical trials can start only when you perform preclinical toxicology studies. Preclinical toxicology study means the finalized formulation will be injected into rodent, non-rodents in a different routes with a different concentrations, and you will be looking for adhesion parameters and histopathology and hematology. All these things you will check for the safety of the antigen. If it is passes this stage, if the vaccine induces immune response and it is found to be safe when it is being administered in different routes with a different concentration, especially higher concentration than the one which you are using for clinical trials, then vaccine will go to the next level. Of course, this will cost activity, and it will be done with the ceremonies. Next slide. And now CSO made a compulsion that every vaccine which you are going to give it to the women's childbearing women, it has to go through DART. They call it as a development and reproductive toxicology tests. Apart from regular PCU, it has to go through DART assessment, where you will be vaccinating males and females with your finalized populations. Then you will allow them for mating, and you will evaluate the vaccine impact on pregnancy, fetus development, and embryo development, and even at the lactation period, how litters are behaving. All that study needs to be performed when your vaccine or when your target population includes women in the vaccination. So this is a quite expensive study. Again, it has to be done by GLP certified laboratories, which may cost around crores. Next slide. So then comes after this, you will go to the subject expert committee that usually have doctors. They will evaluate your preclinical toxicology study, your DOT study, all CMC, everything they will analyze, they will test. Then they will say, now you are ready for phase 1 study, phase 1 assessment of your vaccine candidate. Next slide. So when it comes to phase 1, usually the vaccine testing will be done in the healthy subjects, okay, with the 10 to 100 numbers. I would say 10 to 100 numbers who are very healthy subjects, okay. The primary objective of the phase 1 is you will be looking first safety. You are not going to check for the protective efficacy or immunogenicity. First thing is safety. That is the primary endpoint in the phase 1. You will administer. That's why you are going to use healthy subjects. Subjects who are healthy between, like most of the time, you will be adults between 18 to 59 days, and you will be looking for the safety, adverse events. Then comes the secondary endpoint as an immunogenicity. These 2 points, if it passes, then again we present this data to subject subject expert committee to take the approval for phase 2. In phase 1, there is one more possibility that you have done a dose escalation. For example, 5 microgram, 10 microgram, 20 microgram, which showed better response in the preclinical studies. You don't know which one works better similarly. Then you need to do dose escalation studies. Okay. So phase 1 will tell you next step. Vaccine is safe. It is not causing any severe adverse in humans, especially in healthy subjects. Then you will also come to know which dose is optimal. With that, when you submit that data to CDSU or the CC committee, you will take the approval for phase 2. Phase 2 is it's more or less phase 1, where the number of subjects will be more. It will be usually, I would say, 100 to thousands. Okay? So phase 2 study will be performed with a finalized dose, finalized formulation, in again healthy subjects with a more number of subjects. Okay? Here primary endpoints are immunogenicity as well as safety and adverse events. So once you perform the phase 2, you will go to phase 3 studies. Of course, phase 3 is the final study for vaccine development, where it will be done in thousands to 100,000 population, which covers most of the risk groups as well. Except kids, it covers all age groups, and you will be evaluating your vaccine in large number of populations. So phase 3 primary endpoints will be immunogenicity. Of course, safety will be there. There comes efficacy as well. And every clinical trial there will be a placebo arm, which I forgot to mention. That's a control which did not take except active ingredient. The entire formulation will be the same. And most of the time, most of these studies happens in a blinding condition. Means sponsor as well as the patient don't know whether they received vaccine or placebo. There will be a blinding process. After performing the activity, they will unblind and they will see the response, adverse events, even safety aspects and efficacy. So that way it will be calculated what is the efficacy of the vaccine by seeing the incidents. For example, in COVID case, what they did, when COVID was evaluated in 30,000 population, 2:1 ratio of the vaccine versus control. So they looked into the COVID-19 cases in control as well as the vaccine groups. Then they calculated the efficacy. That's how we will be perform the vaccine efficacy of the next one. So with the phase 3 data, you can apply CDSO for marketing authorization. That will allow you to, and phase 3, one more thing will happen. Industry has to manufacture consistency batches. Okay? So you need to show that when you are producing the vaccine in a 10,000 liters day in a different batches, those batch derived antigens will be formulated in phase 3 and show that there is a consistency. With that, phase 3 will be done, you will go for marketing authorization, your vaccine will be launched in the market. Then comes the phase 4, it is called post-marketing surveillance. Most of the time, it happens in those MNC companies. After launching the vaccines, they will start surveying the recipients. It is similar to phase 3 trial, but it happens with a larger number. Okay? And there is a possibility that in post-marketing surveillance, they may see different scenario which was not observed in the phase 3, because this is actually called as a real-world scenario. They may see new events which were not occurred in the phase 1, phase 2, phase 3. And there are some extent that phase 4, they see deaths, they see severe adverse events. So these are the 3 examples, different examples which I have mentioned here. One is a dengue, which got resuspended Brazil very recently. There is a vaccine from Bhutan, okay, which was phase 3 was done in 60 countries with 29,000 subjects. Phase 3 showed no adverse events, vaccine was fast and approved. Now, in 1 year, Brazil started ANVISA, there is a regulatory body in Brazil. They approved this vaccine in Brazil population. In a recent, they found that 2 deaths and 48 severe adverse events. With that, they suspended the vaccine. Similarly, there is one more vaccine, FDA suspended this vaccine in the US, same case. This vaccine is against chikungunya. So these things also happens after when you invest so much and when you produce the thousands liters of antigen and do marketing and after enrolling, after administration. And these are not only suspension of that particular case. With this particular incident, company will face a lot of negative. Of course, there is one inclusion from our other COVID-19 vaccine. Vaccine was also suspended by WHO when they came and they saw the facility. They said GMP is not meeting our criteria, hence this vaccine, because WHO approved this vaccine for other countries, so they also said. So phase 4 or post-marketing surveillance depends on various reasons. This is the pathway of vaccine development and testing, enrollment, and again there is a post-marketing process. So with this, I like to say vaccine development is a real challenge for manufacturers and the regulatory authorities to evaluate and to approve that. At the same time, inventors who develop the vaccines, where they take decades of decades to find out functional antigens, later they come into something else. So variability, global relations also contributes, and the scaling up is a big problem. All these things suggest that vaccine development really a big challenge for manufacturers. So with this, I would like to conclude my presentation. I'm grateful to organizers and Senator Kovani Happy, and I congratulate Senator Minister Darish. Thank you so much.
Yeah. Any questions from the students and audience? veterinary point of view, what are the major vaccines you are presently looking for?
Well, when it comes to veterinary part, there is no difference between animal vaccines and human vaccines. There is a biggest truth, okay? Because ethical committees and all regulatory bodies, they treat animal vaccines also same. It goes through similar pathway which we for humans. Of course, postal there is a difference, but it will be going through the same. So Indian hospitals as known for effectively tuberculosis disease vaccine, which developing since 1980. Of course, we are the largest producer, and we also have a vaccine for rabies vaccine, and we do have a canine vaccine for dogs, and yeah, that's the portfolio of vaccines which we have.
So in the short period, there was a invention of this COVID vaccine. So really, it was a wonderful job. So congratulate all the scientists who were involved in So I would like to ask one question. So COVID-19 emerged in the year 2020, but the HIV is there long back. Why there is no vaccine for HIV? Why?
For malaria vaccine, it took 40 years. For pandemic, it took less than a year. This is clearly indicates situation, emergency, and the impact or what you call impact of the disease. Unlike pandemics, HIV, these are all not drastic. Of course, now there is a silent impact is there, but not like COVID-19. Pandemics are different. Pandemics will be treated in a very different way because that not only kills the affected people, it also kills the entire global this thing in a different way. For example, because of COVID-19, there was a shutdown. There is a lot of things in logistics. See, imagine everything was shut down. You can't even transport the essentials. So because of that, COVID-19 or any pandemics, now we were also making some. So regulatory bodies or policy makers treat these pandemic diseases in a different way compared to regular diseases.
So I have a question. When it comes to the market for vaccines, like, how about US regulations and for Indian launches? And like you have experience working with Bharat as well as Indian launches. So how the US market strategies of US market for Indian launches?
See, I would say compared to US, EMA, European Medical Agency is most into that because they do each and every documents of each and every phase of the vaccine development. Both FDA and EMA are stringent in that. It's not like it's an Indian vaccine whether outside or not. You provide necessary documents and if your facility goes through the inspection and passes, I don't think there is an issue. Because in fact, co-vaccine was initially there was a tech transfer, the US company started working on that. But when WHO suspended this, then there was an impact on it. So that way, they're looking vaccine alone, vaccine candidate. Because see, co-vaccine was an inactivated vaccine. That is the conventional platform. And they use the potential antigen. That way it was a very novel vaccine. They respected that.
Questions? If there are no questions, I'm glad to take the session of Dr. Raju, invited all, the Vice President of Indian Immunological Unit in Hyderabad. So he has covered in his talk bench to field challenges and regulatory considerations in vaccine manufacturing. So I'm glad to the co-chair Janara guidance also. So he has covered in detail about understanding the vaccine development process and also the COVID-19 vaccine development, different regulatory approvals and hurdles for vaccine development, and key aspect of selection of antigens for vaccine development and vaccine development key elements of serious covered. And also the upstream and downstream process for the production of vaccines and various technical issues for preclinical and regulatory toxicological studies also. And in detail, he has covered the clinical trials of the phase 1 to phase 4. So in very fantastic way, very excellently, he has about vaccine. Thank you, sir, for your nice talk. So I think this is positive to hold.
ಇದು ಸ್ವಲ್ಪ ಸೈಡ್
Thank you, sir. Now I request Dr. Rajeshwari ma'am to facilitate general procedure. Thank you, ma'am. Go to the next session. Before that, we are having one more session. I invite Dr. Jubilee Deshpande Rao, Scientist C, ICMR, Puri, for the next session. And I call upon Dr. Manjunath Sargi sir as a chairperson, research scientist, Haskell Institution of Medical Sciences, and Dr. Chaitanya sir as a co-chairperson. Now, on behalf of Dr. Jubilee Deshpande Rao sir, Scientist C, ICMR, Puri. Over to you.
So now, good afternoon to all of you. I would like to introduce another allied person, Dr. Jubilee to this session. Sir is working as a scientist. He is as well as assistant professor, ACSR, surveillance, epidemic and preparedness group, and Resource Center for Virus Research and Diagnostic Laboratory Network. And research expertise is main area of research is humoral immune responses in viral infections and vaccinations, and development of serodiagnostic reagents method for viral infections, monoclonal antibody development, laboratory quality control, quality assurance, development of reference standards, external quality assurance standards, and quality control material for viral diagnosis. And also, sir is also involved in translational research and laboratory capacity building. And his major contribution to brief, he has led the government of India, Iran mission during the COVID period for safe expatriation of more than 500 Indian national standard in Iran during COVID-19 and translational research that includes lives of TRND, MNP, anti-SARS-CoV-2, ING, ELISA, and whole cell antigen. All these assays were used in vaccine preclinical and clinical trials, vaccine efficiency testing also, and national sero survey. And understanding the environmental immune response among co-vaccine, Covishield, and Sputnik vaccines. So, as a human resource development, including so your resume curriculum like is very huge. So I am not able to complete it within 5 minutes.
Thank you.
So and sir also involved in important vaccination trial and awareness and also Iran project. So I would like to invite Dr. Deshpande sir to the audience. So please take on this session.
Thank you very much for the detailed introduction. It is my privilege to be in front of you all as a part of this unique group to share the story, which is not a scientific discussion what I am going to drive because it is a lunchtime and it's very difficult to digest a lot of information. We ourselves call as a white collar soldiers. So it's a huge team involved in kind of pandemic and epidemic preparation. But a part of that journey, what all things are happened and how exactly behind the scenes work to develop a capacity to the country of this population. So this kind of story is there when there is a pandemic or epidemics are happening. I would like to share that story through this journey. Next step. So if you look at the diagram, there are so many marks which I am able to see. Each marking is indicated one of the other type of viral outbreaks which are happening since from 1950s to till date. During that 1950s to 2013, only 2 institutes were capable of handling viral infections in the country. Only the first institute is ICMR, National Institute of Virology, and other one is the National Center for Disease Control, which is situated at Jubilee. So by the time the sample reaches to that institute and then the report goes back, it used to be 15 days to 1 month time period. By the time the pandemic or the outbreak situation used to be complete, and then the spread of infection is also one of the major concerns. Because of that, ICMR headquarters decided to have a group of laboratories which can able to give the diagnosis and then provide the diagnosis within 24 hours of time so that we can contain the disease spread. Next slide, please. With that thought, ICMR started developing centers called viral diagnostic laboratories. Starting from 10 laboratories, now we have 165 laboratories all over the country. You just see the Indian map. Every corner of the country, each state has at least 5 to 6 viral diagnostic laboratories. After some days, when the realization happened, why diagnosis doesn't help, we need to also pursue some level of research in them to maintain the sustainability. So because of that, research component was also added, which is called now as the virus research diagnostic laboratory network. And there are 3 layers of networks you can see. One is medical college level, second is state level, and regional level has the highest type of laboratory, which has the minimum facility for routine diagnosis of public health important.
Next slide. So mandate is to just give the routine diagnosis and also provide the outbreak response whenever there is a huge number of cases are happening in one particular place. They also provide investigate and also provide support for the epidemic family. And also include in this multi-scientific studies whenever there is a policy is required to develop the evidence. So also develop the training and research facility so that the medical college level staff can also train. Next slide. So this is the infrastructure what they have. Regional lab has the biosafety level 3 laboratory along with this cellular diagnosis and then molecular diagnosis. And state level has serology as well as molecular diagnosis, and then medical college also has this. State level additionally has the tissue culture. Next slide. So these are the viruses which they have to test routinely and report to the tertiary care hospital so that routine diagnosis is provided free of cost on the public health system. And if there any new cases are coming which are not actually reported routinely, is picked up and then the containment can happen. So that is the testing capacity what we have for this. So these are the stakeholders. All these PRDLs are interacting with all of them. That's why anywhere any new cases are coming, any new viral infections can be easily picked up, which includes private medical colleges, government college, and then national programs like vector-borne disease control networks. Next slide. So organization-wise, Ministry of Health is handling DHR and ICMR. Through that DHR ICMR, we have two research centers. One is situated at ICMR in Pune, where I am working, and the data management is happening because the huge data which is generated is being monitored, and then that actually gives the real-time evidence of what kind of virus infections are happening in the time. And 3 layers of labs we have that I already mentioned that in the state and the medical part. So after looking at the huge infrastructure what we have built up as a country to capacity for viral infections, there was a need of research center to provide all the developed and human resources who can able to easily test the viruses in different sites by using the tools. Second is quality control, because testing has to happen in the right way. If you are giving a wrong report, false positive, then it is an issue that represent a false signal to the country. That's why the quality control system has to also be established and scientific and technical support day to day. That is another mandate of this research center. This research center what we are presently handling so that we can able to achieve these objectives and build the capacity for the epidemic. So like this, we have a set of guidelines which are readily available for all these laboratories, which includes list of viruses, testing algorithm, testing strategy for viruses, ELISA kits, frontline tests, and then clinical history. All of these actually used in the guidance. So uniformity throughout the country is maintained to this This is also provided to all these laboratories. So along with this, we have some more resource materials that have taken for the training. You can see that more than 6,000 people are trained. And the 18 viral parameters have been tested every year, more than 7,000 to 8,000. Along with that, we are maintaining the quality control for all of these laboratories. So these are the different activities, how we support to them. Being in Pune and providing all the support all over the country, so that they are ready available whenever there is a viral disease outbreak, anything like COVID pandemic. So next slide. So there are 3 types of training programs we conduct. One is the regular routine training program for viral diagnostic, diagnostics for the routine testing and providing the support to the public health system, the tertiary healthcare centers. And second is emergency preparedness program. Recent news which is spreading Ebola cases are increasing in the other part of the world. So for that purpose, we are preparing our country capacity for diagnosis of such kind of emerging infections, and then national programs to develop the evidence. Whenever there is a vaccination program which is to be established and then provided to universal immunization program, we have to create an evidence how this vaccine works and the immunogenicity part and the capacity of the
Zika, COVID-19. So such kind of emerging viruses are coming in the global scenario, and during that time, beforehand we will prepare the labs, diagnosis and prevention. Along with that, quality control, I said, this quality control program actually monitors the quality of testing in all the laboratories, so that similar kind of quality has to happen, so that correct results will reach the ready in time. So overall the capacity what we have reached over the years since from 2013 till date. So now how it is actually being used in the academic and pandemic situation. And just want to example, next slide. Taking an example of Zika virus outbreak which has happened in 2016, when there is a huge outbreak, more than 150 countries in the South American area, they got infected. And during that time, Asian Games were actually ongoing. Sports persons went there for sports participation. When they came, they were coming back. Government of India told us we have to test them for the Zika positivity in case because of the test. Zika actually causes the congenital disorders as well as microcephaly conditions. So because of that, there is also there is a Guillain-Barré syndrome. So because of that severity which is observed in South American countries, so we had to establish the testing facility and then test the response person sample and then make the report. So along with that, we established the network of laboratories utilizing the BIOTECHNOLOGY platform. So we trained initially 10 labs and followed by 132 labs. Because of that, for the first time, we identified the first case of Zika in the Ahmedabad state. Ahmedabad, which was when we looked at the clinical case sheet, it was surprising that case is within the country. It was not like any history was not there in the endemic area. They have looked at bottom country. That indicated that the Zika virus is silently transmitting in the community. So that was the one first report laboratory evidence created by the surveillance network, and then followed by that in 2018 for the first time the huge outbreak was identified for Zika through this BIDL network. So like that, in that process we isolated Zika virus and then the characterized and developed the reagents required for the diagnosis. So these are a few examples like mononuclear antibodies we generated to develop the serological diagnostic methods so that indigenous methods are available and they are cost effective for the country. Next slide. Next slide. So in 2024, one outbreak was by innate cell. In that case, we investigated and identified such isolated strain which is not actually causing the more pathogenic conditions when we look at the Brazil isolate. So that was actually giving a level of confidence and then answer for the public health because of microcephaly involvement and congenital disorders. So this strain was not causing any kind of such congenital disorders. Next slide. So that was about the academic preparation and then disease surveillance program. How quickly we can able to establish things we have the network practice which is readily available with the capacity. Now that taking COVID-19 as an example, pandemic situation, you just look at the first before January 18, 2020, the first case was actually reported in India in February. Before that, we started our activity. In that month of January 2020, only one lab, only NIV was used to be a testing center. As the number of cases increased globally, within a 1 month time, we established all the testing facilities through that training program. Quick action actually helped us to have the labs around the India, so who can able to test this COVID-19 report quick. Otherwise, next thing. Containment facility was very limited and was very challenging. In the meantime, next slide. So there was one assignment also provided to us as a part of the pandemic requirement. Iran mission was launched by Government of India. I was fortunately a part of this program, and more than few thousand of people, those who actually stranded because of the international border closure, we brought them back safely to this mission. Next slide. Next slide. So along with this, private agencies also participated in the meantime. Everything was monitored at one center through ICMR and Pune, and then disseminated all over the country. We were actually part of that team, and that was one good opportunity, starting with the 13 BIDLs and now 165 BIDLs. along with the private agencies. So next slide. All of them they proved that the country can be actually easily handling such kind of huge pandemic by providing the diagnostic support, by providing the right time within short time period of patient care and all. So next slide. So along with this, there were multiple activities we also pursued. I was involved with the serological assay development. So this assay development was the key requirement for the overall disease burden assessment. We developed an IPC ELISA which was very much required at that time to understand the total burden of the disease, which is spread all over the city in different areas. Based on that disease burden, there was a kind of color coding in the municipal corporation. So that is why this kit was developed and transferred to the companies, and then they produced, and this was used in the national zero survey program. Next slide. So next question came in the meantime. How long the immunity, once you get exposed to the SARS-CoV-2, how long you can able to have an antibody? Next slide. We quickly developed one ELISA which can detect the IgG level for spike protein, the immunogenic protein for SARS-CoV-2, and then established that up to 8 months period the antibodies are still there, and 80% of people they actually retain the protection. Next slide. So next question was, we have an antibody present only but it is not functional. Is it viral? Virus can be stopped by replicating. So functional antibody testing platforms were required at that time. Next for pre-clinical studies, vaccine trials and all. We quickly developed the neutralizing platform for SARS-CoV-2, and then those platforms were used for vaccine studies and pre-clinical studies. Next slide. So since talking about medical co-vaccine, so behind the scenes, what we all did the effort as a part of the initial preparation. So actually, we made a platform for me to introduce and explain that different stages where ICM or NIV pooling was part of the vaccine development, starting from isolation of the virus and characterization of virus, followed by the optimization of the neutralizing assays to understand the preclinical studies to perform in the small animal models and then larger animal models, and then we went ahead for immunogenicity testing, and along with the therapeutic we did the test 1 and test 2 trial, and we moved to the test 3. So that actually led to a readily available product with a lot of effort from the biotech company as a co-vaccine product. Next slide. So these are the initial studies just to establish immunogenicity of this particular product is there in the small animals, followed by regulatory agency also wanted to take clinical studies in the non-human primates. So we also did that study. We were also part of that study. That was a major assignment in the COVID-19 pandemic. Next slide. And there were two phase publications you can able to see now. They are part of this phase 1 and phase 2 trial published by BIOTECH. We were also involved in that immunogenicity testing and quality control testing. Next slide. So next slide. So next question came. These are all public health questions. People used to ask us, and we used to develop an evidence. Do co-vaccine and co-vaccine. By that time, 2 vaccines were approved for the vaccination for the public, and there are a lot of variants which were emerging continuously globally. People ask that different variants are coming, and co-vaccine and co-vaccine are of earlier platform. Once they type, are they protecting against the new variants? For that also we generated evidence by performing these vaccinated sera against the different variants neutralization test, and it was very much proved that with the different fold degrees, definitely there is a level of protection still remains that we proved with the latest strain. Next slide. So with the upcoming new strains at that time, beta and delta strains. Next slide. So then also we also look at primary dose, secondary dose. Is it 1 dose is sufficient, 2 dose is sufficient? Again, another public health question was there. For that also we generated evidence. Based on this evidence, Government of India made some policies at that time. So next slide. So next slide. Next question was, there was one case in Jubilee. By mistake, one nurse gave a dose of Covaxin who has taken Covishield in the first dose. So that a lot of news was there. At that time, ICMR decided to have quick cohort study by providing heterologous vaccine. 2 times first vaccine, later he or she can get a Covishield or not. So like that, that kind of cohort study was conducted, and we answered that there is a robust immune response. We can able to see, even though there is a change in the platform, because the COVID vaccine is the inactivated vaccine and the Covishield, as you all know, it is a different kind of platform which has, so which came from the UK farmer. So this indicated that robust immune response as well as the safety factor, and then people started utilizing this data, and then later government allowed for the heterologous vaccination. Next, another question after some time came, everything was settled, and then people used to get reinfected multiple times. So one infection, they got both full dose, and then again they used to get infected. At that time, people asked, "Whether the vaccine we have to do the infection over?" So booster vaccine was the another concept. People came up with that question. Is that booster vaccine really required? So third dose is required. For that also we established an evidence again, and that evidence helped us to show the evidence to government, and then government allowed for the third dose introduction in the public. Next slide. So this is about the pandemic preparation and how during the pandemic ICMR National Institute of Virology actually acted as part of the national preparedness program. So by both for epidemic, this local outbreaks as well as global pandemic kind of situation, the silently we are preparing our country its capacity through this BRDM network, and that helps a lot. So in the absence of both epidemic and pandemic, what we do multi-scientific studies to create an evidence for public health policy making. For example, now the question is, Dr. Raju mentioned that dengue vaccine was suspended in one of the countries. So now we need to have a dengue is endemic to our country. We need to have a vaccine in place. So because of that, who will do that regulatory trial? We have to perform our own so that we can see our population vaccine is acting properly and safe, and then bring a robust immune response. That's one major activity we are doing. Along with that, Government of India, along with WHO, is targeting measles control, elimination, and then rubella control program. That also we are performing because the measles and rubella vaccination is a mass vaccination program, which is there in the universal immunization program, and like new emerging infections like monkeypox diagnosis, along with the other team at ICMR and NIP, we are working through this network to provide a diagnosis and contain the disease, so that the disease is only contained in particular area where the disease is also initiated. Otherwise, because of the huge population, there is a huge outbreaks which are expected to happen. So like these multiple activities we are performing as a part of the capacity building program. Along with this, we also involved in the outreach program so that sustainability to bring that science motivation to the young children, which actually helps us our country's major vision of our Prime Minister. So to achieve the strength of our country, we need to have initially we have to motivate our youngsters towards science perception, and then do in-depth R&D so that we can have our own products which are indigenous and faster. So this is the campus. We have 2 campuses in the ICMR, and you can visit those who are interested to know more about viral diagnostic laboratories and also virology institute, and opportunities for both all life science students, biochemistry, botany, zoology, biotechnology. So there are multiple opportunities through this 165 years. As I showed, there are different opportunities for scientist positions, technical assistant positions, and research positions. You can explore that opportunity through this. So as the door goes, Guru Prasad Siddhi means the success is attained through the blessings of guidance of a Guru. So I dedicate all my work and presentation today, and then the earlier one what I did, that motivation came from the blessings of our teachers. Special thanks to Dr. Chandrakant sir, who motivated me and allowed me to go in this R&D journey to this guidance, eminent guidance, and also all the teachers who made me what I am today. So I dedicate all my work, and the future. to all my members. Next slide. So this is our team at SMRNI unit. They work in different aspects. Some of them are in the training program, some of them are in R&D with me, and they are the major support for check. Thanks. So many questions are there. I'm very happy. I'm sure that there will not be any more questions because of time. And they are very enthusiastic and any quick questions?
If no question from the audience, so I would like to congratulate Dr. Guraj Deshpande sir for his immense contribution to the nation building as well as health and wellness of the people during the pandemic period. So he has also worked as a corona warrior during the COVID period also. Now I would like to hand over this mic to chairperson of this session, to Manjuna.
I am very glad to say a word. First of all, I am a part of this VRL method. Actually, I am very happy what exactly the RC VRL is provided training to all the VRL from starting, from initial days, from nobody was ready to do testing, from then now entirely to the next level of testing we can do in the entire pan-India. That is immense effect once done by RCVRL, that is running. Through that, I should be congratulating the other team like DHR and everything. Thanks for the audience. Thank you.
Thank you, sir. And now I request Dr. Param sir to felicitate our speaker, Dr. Guraj Deshpande sir. Now I call Sardar Hussaini sir to felicitate Manjunath sir. Dr. Manjunath sir. Sardar Hussaini sir. Now I request Dr. Sudhi Reddy sir to felicitate Jayachandra sir. Sudhi Reddy sir to felicitate Dr. Jayachandra sir. Thank you, sir. Thank you, sir. Now it's the lunch break. You can assemble again for 8 to 15 p. Thank you.
ಬಂದ್ ಮಾಡಿ